A harmless chemical reaction can destroy a $150 million survival machine. It begins inside wet concrete. No alarms detect it. No computer identifies it. Yet it can slowly drain oxygen from human lungs. In 1991, engineers sealed a glass fortress in Arizona. They called it Biosphere 2. The structure contained forests, farms, oceans, and living ecosystems. It was designed as a miniature Earth. The mission was simple: prove humans could survive inside a closed world, then apply that knowledge to Mars. 8 scientists entered the structure. The steel doors locked behind them. Advanced computers tracked temperature, pressure, water, carbon, and oxygen. Every valve had a purpose. Every airflow followed a programmed route. Nothing important was supposed to escape. But after several months, something began removing oxygen. The decline was slow. Then it became impossible to ignore. Breathing required more effort. Simple work became exhausting. Short-term memory began failing. Some crew members woke during the night, gasping for air. So, where was the oxygen going? The glass remained intact. The steel shell showed no major leak. Plants still received sunlight. The machine still reported normal operation. Yet the atmosphere kept thinning. This created a brutal contradiction. Biosphere 2 was built to preserve life. Instead, it was quietly pushing its occupants toward collapse. The more tightly engineers sealed the system, the more dangerous it became. So, what happens when history's most expensive survival machine suffocates internally? What happens when the enemy has no moving parts? And how do you stop a failure hidden inside walls? The answer is simple but brutal. This was not an ordinary biological malfunction. It became a forensic materials investigation. Every ecosystem became evidence. Every pressure reading became a clue. And somewhere beneath the glass, a silent reaction was operating. It had been active since the doors closed. The failure had already entered the structure with its foundations. To find it, investigators first had to eliminate every obvious suspect. The first suspect was the structure itself. Biosphere 2 was designed like a sealed mechanical womb. Glass formed the visible shell above the Arizona desert. Thick steel plates formed another barrier underground. Engineers welded those plates into one continuous defensive skin. Air could not slip through the soil. It could not escape beneath the foundations. Every joint had to hold. Every seal had to survive constant pressure changes. But a sealed world creates another problem. Sunlight heats the interior air during daytime. Cooler nights force that same air to contract. Without pressure control, the glass could bend or crack. So engineers built 2 enormous mechanical lungs. Each lung absorbed the daily expansion and contraction. Flexible chambers moved as the atmosphere changed volume. They protected the shell without opening it outside. Airflow followed programmed routes through the complex. Fans moved oxygen across forests, farms, and living spaces. Sensors tracked pressure throughout every connected biome. Computers watched the atmosphere like a permanent security grid, detect the leak, localize the weak point, measure the loss, then seal the breach. But the readings showed no catastrophic opening. The glass remained sealed. The buried steel barrier still held. The mechanical lungs moved exactly as designed. There was no obvious path outside. Yet oxygen kept falling inside. So the fortress was not leaking air normally. It was losing life through another mechanism. If the shell remained intact, what failed next? The answer seemed biological. Plants produced the system's replacement oxygen. If production slowed, the atmosphere would weaken. So investigators turned toward the living canopy. The forest became the primary suspect. Plants were Biosphere 2's biological oxygen factories. They absorbed carbon dioxide during daylight. Then photosynthesis released oxygen into the sealed atmosphere. Those factories stopped, the numbers made sense. Oxygen would fall without any physical leak. So scientists activated the environmental sensor network. It became a patient biological search system. Instruments scanned the artificial rainforest sector. Others tracked the savanna and cultivated zones. They measured light, gases, airflow, and plant activity. The search moved through every green compartment. Think of one careful finger crossing an ocean floor. It tests each surface without skipping hidden spaces. Investigators looked for failing leaves. They searched for damaged roots and blocked photosynthesis. They checked whether plants still captured carbon dioxide. They checked whether sunlight still powered oxygen production. Arizona sunlight continued striking the glass structure. The trees continued responding during daylight. Leaves still absorbed carbon dioxide. Vegetation still released oxygen under available light. The biological factories were not completely offline. They were operating inside their designed role. But the total balance remained wrong. Oxygen production could not match oxygen consumption. Something else was pulling harder than plants could replace. So investigators peeled away another layer. The threat was not floating above the forest. It was not hiding inside the glass canopy. It was operating beneath every root. The soil looked passive and harmless. But soil is never chemically silent. It contains living populations that also breathe. Inside Biosphere 2, conditions were no longer normal. The next suspect existed everywhere. The soil carried unusually rich organic material. That material was meant to support rapid plant growth, but it also fed an invisible population below ground. Bacteria began consuming the available organic matter. Their activity expanded across the sealed ecosystem. It was a distributed biological swarm. Every particle became another feeding zone. Every feeding zone demanded more oxygen. The bacteria inhaled what the crew needed. Then they released carbon dioxide as metabolic exhaust. Individually, each organism consumed almost nothing. Together, they formed a system-wide oxygen drain. The attack came from beneath every planted surface. It did not break pipes or damage seals. It simply used normal biological respiration. Nothing appeared mechanically broken. The microbes functioned exactly as biology intended, but their combined scale overwhelmed the closed atmosphere. Oxygen moved downward into the soil network. Carbon dioxide should have moved upward into the air. Plants should have captured part of that carbon. Sensors should have detected the remaining increase. This created a clear working explanation. Rich soil activated the microbial swarm. The swarm consumed oxygen continuously, but the explanation still contained a weakness. Carbon dioxide did not rise enough. The bacteria were releasing enormous quantities, so where was that gas accumulating? It was not leaving through the sealed shell. It was not fully returning through plant growth. Yet sensors could not find the expected volume. One mystery had solved the oxygen loss. Another immediately replaced it. Still, the oxygen decline continued. Time was now disappearing. The atmosphere was approaching a human performance limit. The failure moved into human bodies. Oxygen eventually reached 14.5%. That level changed every ordinary task inside. The crew now lived under high-altitude conditions. Their bodies behaved like they occupied 4,000 meters, but no oxygen tanks waited inside. Walking required more effort. Physical work drained energy faster. Some crew members developed sleep apnea symptoms. Their breathing repeatedly stopped during rest. Sleep no longer restored normal performance. Short-term memory weakened. Midnight exhaustion became part of daily operations. Red blood cell levels rose sharply. More cells meant greater oxygen carrying capacity. But adaptation could not rebuild the atmosphere. It only helped bodies endure the decline. The experiment had reached its physical choke point, so management faced a direct choice. Protect the closed world principle, or protect the people living inside it. Both goals could no longer survive together. The answer was simple. Human survival came first. Liquid oxygen from outside entered Biosphere 2. Breathing conditions improved. But the mission's central promise had been pierced. This world was no longer fully self-contained. Its survival now depended on outside intervention. The fortress remained sealed in appearance. The biological system had failed in practice. Yet this emergency measure solved only one problem. It restored oxygen without explaining the missing carbon dioxide. The bacteria had consumed oxygen. Their respiration had produced carbon dioxide. That gas should have filled the atmosphere. But sensors never recorded the matching increase. So the largest clue was not oxygen. It was the carbon that vanished afterward. Where did that carbon dioxide go? The answer was not hidden among leaves. It was not buried inside soil. It was waiting inside the architecture itself. That architecture was not passive. It was the missing carbon reservoir. To understand why, investigators had to cut through Biosphere 2. The real choke point sat beneath every visible ecosystem. Thousands of glass panels formed the structure above ground. Those panels needed a massive load-bearing frame. That frame required foundations and buried dividing walls. Engineers selected concrete for both jobs. They poured 110,000 cubic meters. It supported the glass and separated the internal environments. It also touched the sealed atmosphere across enormous surfaces. Concrete appeared strong, stable, and chemically harmless. That assumption became the system's deepest vulnerability. The building was not surrounding the experiment. The building had entered the experiment. Every wall became part of the carbon cycle. Every foundation became part of atmospheric chemistry. Yet the computers treated concrete like dead mass. They monitored air, soil, plants, and human metabolism. They never expected the architecture to participate. But why would concrete affect oxygen? The answer begins before concrete becomes fully cured. Fresh concrete does not instantly become chemically silent. Its internal compounds continue reacting after placement. Biosphere 2 contained concrete that had not fully cured. Then engineers sealed it beside humid artificial ecosystems. The rainforest maintained moisture throughout the interior. Water reached exposed surfaces and microscopic pores. That moisture kept chemical pathways active. The concrete began behaving like a slow chemical lung. It did not inhale oxygen directly. It captured carbon dioxide produced elsewhere. Microbes consumed oxygen while decomposing organic material. Their respiration released carbon dioxide into the sealed world. Under normal conditions, that gas would remain measurable. Plants could absorb some during photosynthesis. Sensors would detect the remaining atmospheric increase. But Biosphere 2 contained another carbon sink. Carbon dioxide reached concrete surfaces continuously. Then the material removed it molecule by molecule. No alarm threshold was crossed. The process looked like ordinary material aging. The mechanism is called carbonation. Inside concrete, calcium hydroxide remains chemically available. Carbon dioxide enters through microscopic pores. Moisture allows those compounds to meet and react. Calcium hydroxide combines with carbon dioxide. The reaction creates calcium carbonate and water. Calcium carbonate is essentially limestone. While carbon dioxide entered the walls as gas, it became solid mineral inside the concrete. The building was manufacturing stone from its atmosphere. This was the missing pathway. Soil bacteria released carbon dioxide after consuming oxygen. Air currents carried that gas through Biosphere 2, but it never accumulated at the expected rate. Concrete intercepted it before sensors saw the full increase. The walls became silent carbon collectors. Foundations became a distributed chemical trap, but the total concrete volume changed everything. One small reaction repeated across massive areas. A harmless process became a system-level failure. Concrete did not create the original oxygen demand. The bacteria did that, but concrete erased the evidence left behind. Oxygen disappeared. Carbon dioxide should have replaced it. Instead, carbon dioxide became limestone. Now the complete chain becomes visible. Rich organic soil fed an expanding microbial population. Those organisms consumed oxygen across every planted zone. Their respiration generated carbon dioxide as expected. Air currents carried that gas toward plants and structures. Plants recovered only part of it. Uncured concrete captured another major share. Carbonation locked that carbon inside foundations and walls, so the biological cycle could not close. Oxygen left through microbial respiration. Carbon dioxide failed to remain available for complete recycling. The sealed world was transferring carbon into architecture. That created a one-direction chemical pipeline. Air became microbial exhaust. Exhaust became mineral. Mineral remained trapped inside concrete. The cycle ended at the wall. Their models separated biology from construction materials. Plants belong to the living system. Soil belonged to the living system. Humans belong to the living system. Concrete belonged to the container. But inside a sealed environment, that separation was false. Every exposed material exchanges matter with the atmosphere. The concrete appeared structurally passive. Chemically, it was operating continuously. This also explains the contradiction inside the mission. It prevented ordinary atmospheric leakage. The mechanical lungs controlled pressure changes. Yet that success intensified the internal chemistry. No outside atmosphere could dilute microbial demand. No fresh air could replace the missing oxygen. No external carbon cycle could balance the system. The tighter the structure became, the stronger the imbalance grew. Biosphere 2 had sealed itself against external failure. It had not sealed its materials against internal participation. That is the central reversal. The fortress was not defeated by a broken barrier. It was defeated by an active barrier. Concrete held the structure together. Then concrete helped pull its atmosphere apart. The same material provided strength and chemical instability. Rich soil supported plant growth. Moisture supported the artificial rainforest. Concrete supported thousands of glass panels. Airtight construction protected the closed ecosystem. Together, they formed a failure network. That network operated below ordinary perception. Microbes consumed oxygen without visible damage. Concrete captured carbon without visible movement. Sensors recorded symptoms, but not the hidden transfer. The crew experienced the consequence inside their bodies. Reduced oxygen weakened sleep, memory, and physical performance. Outside oxygen restored breathing temporarily, but it did not erase the engineering lesson. Investigators had to redefine the entire system boundary. Biosphere 2 was not only plants and animals. It included soil, water, glass, steel, and concrete. It included every chemical bond storing carbon. Once that boundary expanded, the mystery became solvable. The missing gas had never escaped the fortress. It had changed physical form inside the fortress. Carbon dioxide entered concrete as atmospheric waste. It remained there as solid calcium carbonate. The system buried carbon within its own foundations. So the culprit was not biology alone. It was not architecture alone, it was their collision. Microbial respiration created the carbon flow. Uncured concrete captured that flow. Airtight construction prevented outside correction. Human physiology absorbed the final pressure. One microscopic reaction connected every layer. Soil chemistry reached the concrete. Concrete chemistry reached the atmosphere. Atmospheric chemistry reached the bloodstream. Biosphere 2 became a forensic materials case. Now, one question remains. How can future habitats stop architecture from joining the ecosystem? Future habitats must treat every material as chemically active. Concrete cannot remain exposed inside a sealed living system. One solution is complete polymer isolation. Specialized coatings can separate concrete from internal air. They block moisture, carbon dioxide, and reactive surface pathways. The structure still carries weight, but its chemistry no longer enters the atmosphere. There is another option. Let the concrete cure before sealing anything. That process cannot be rushed. The material may need 3 years in open air. During that time, major reactions occur outside the habitat. Only then should engineers close the environment completely. This changes how survival systems must be designed. Engineers cannot separate architecture from ecology anymore. Walls exchange gases, floors absorb moisture, coatings age, sealants react. Every material becomes another biological variable. So the lesson from Biosphere 2 is not failure. It is forensic discipline. Science does not promise systems that never break. It builds methods for tracing exactly why they break. Follow the oxygen, track the carbon, test every surface, then connect microscopic chemistry to human survival. That process must reach deeper than visible equipment. Must examine reactions inside thick concrete barriers. Even 20 feet of material can hide active chemistry. Strength does not mean chemical silence. Stability does not mean environmental neutrality. A Mars habitat will be far less forgiving. Outside air cannot be pumped through an emergency valve. Replacement materials cannot arrive quickly. One hidden reaction could distort the entire atmosphere. One overlooked surface could redirect the carbon cycle. So future habitats must be designed as complete chemical ecosystems. Concrete, polymers, metals, water, soil, and humans all interact. Nothing is merely a container. Nothing is completely passive. Biosphere 2 proved one final principle. The smallest reaction can defeat the largest survival machine. Humanity wants to build a civilization on Mars. How many invisible traitors already wait inside our blueprints?